Hey everybody, welcome to another edition of the Lab 207 webcast. My name is Mr. Kite. I'll be hanging out with you today as we continue on in our series about genetic technology. The subject for today is genetic technology itself. Now I'm going to warn you right now, we got a lot of stuff to cover today. We're going to go through a bunch of different applications of genetic technology, or I guess techniques that are used. But before we get into all that, let me give you your objectives. Here are the things that you need to know or be able to do by the end of this video. First of all, compare and contrast multiple genetic technologies. For each one, you kind of want to know how it works and what it might be used for. Describe in detail PCR, electrophoresis, and southern blotting. Those are the three techniques or the three technologies that you should really be familiar with. And understand the use of a DNA probe. This is an idea that applies to several different technologies. So all that being said, let's go ahead and start talking about things. So the first idea or concept that you need to be aware of is a DNA library. In a previous video, we had talked about using bacterial plasmids to create tons and tons of copies of a gene. Now, the thing is, researchers would like to be able to keep all of these copies of genes around for use whenever they would like to use them. So they build these things called DNA libraries. And what they'll do is they will take a bacterial plasmid, and they will take the whole genome of an organism and chop it up into a bunch of bits. So let's say we want to make a library out of all of the genetic material that is found in Drosophila, a fruit fly. We would take all that genetic material, we would chop it into a ton of pieces using our restriction enzymes that we had talked about. Each of these pieces would be inserted into a plasmid. That plasmid would then be put into a bacteria that would duplicate over and over and over and over again. Remember, every time that thing divides, you get a copy of whatever gene is in there. Now, this is not a very precise approach. You are cutting out the gene of interest, all of its introns, exons, everything else, and sticking it into this plasmid and then into the bacteria. And then what will happen is a DNA library essentially looks like a big ol' dish that has got a ton of wells in it, and after each plasmid has been duplicated over and over and over and over again within that bacteria will be housed in one of those wells. So they will know that this gene exists in this well, the other gene that they want might be in that well, and the other gene might be in that well. So it's kind of like a shotgun approach, but that is one way to have readily available genes on hand for research. I've got right here BAC. BAC stands for Bacterial Artificial Chromosome. So this approach right here is just known as the genomic approach. You're taking natural plasmids and you're sticking pieces of the DNA into them. The problem is you can only stick small pieces in here. So scientists have figured out how to engineer artificial chromosomes that can be stuck into bacteria, and they are much bigger and they're a little more precise. They don't necessarily contain all the introns and the exons and all the extra bits that you may not need. They are only kind of just the stuff you need to get the job done. So know that is a bacterial artificial chromosome. Now within these DNA libraries, there is a third type that is based off of a molecule called cDNA, that is complementary DNA. Let's say that a scientist wants to get a very nice, neat, precise gene. They don't want the introns, they only want the exons. All they need is the business end of this thing. What they're going to do is they are going to isolate the mRNA that codes for whatever gene they are looking for. They're going to take that mRNA and they are going to cut it up. We're going to make some really ugly little scissors. They're going to cut it up, they're going to cut out those introns and glue the exons back together. So you now have one piece of mRNA that is all exons, and it's got all the information it needs to produce whatever they're trying to produce. They will take that mRNA and then, using reverse transcriptase, we talked about that from HIV, they will use reverse transcriptase to use this mRNA to build a copy of cDNA. So this would go and it would match up, complementary base pairing, except for rather than building RNA off of DNA, we are building DNA off of RNA. They will take this right here, split it in half, and then this DNA strand is single-stranded. It'll be put in a solution with the right enzymes and the proper bases, and it will be fully formed into complementary DNA or cDNA. Our researcher now has got a fully ready to go gene that can then be kept in a library like we just talked about. So we've talked about all of these genes and stuff. You know, scientists have got these libraries of genes laying around, but how do they actually find the gene that they need within that library? And I'm going to warn you right now, there's probably a bad edit in there somewhere. My computer freaked out. Anyway, so how do they find the gene that they need? Well, remember before we get into this that A always pairs to T, C pairs to G, and that scientists have figured out how to artificially engineer strands of nucleotides in the lab. So if, for say, a scientist knows that the gene that they are looking for in this library that contains thousands and thousands of genes has a sequence in it that looks something like this, they can go to the lab and construct a sequence of DNA that would be complementary to it. And what they will do with this complementary DNA is they will actually label it with a type of phosphorus that makes it fluorescent. So all of this might be labeled with a blue fluorescent dye. So what will happen is they can take their artificial DNA, they can match it up, put it into each of the wells of their library, and whichever well has the gene that this artificial strand is able to base pair with will glow. It's kind of cool. It's like saying, hey, here I am, here I am. And other ones will not glow because this fluorescent gene or this fluorescent strand of nucleic acid won't be able to base pair and hook up and become fully functional. So this is how they screen big libraries. It's called a DNA probe or nucleic acid probe, and it uses nucleic acid hybridization because you're creating a hybrid molecule. All right, just a couple more things to cover. In genetic technology, it is great that we can stick the genes of eukaryotes into a bacteria, which is a prokaryote. Problem is, prokaryote and eukaryote gene expression is different, and prokaryotes don't necessarily always have the machinery needed to express a eukaryotic gene. There are several ways that scientists work around this. We're not going to get into all of them, but one of the techniques that they use is sometimes the machinery of a prokaryote doesn't know where to bind on a eukaryote's DNA, because eukaryotic DNA doesn't necessarily have a promoter or a nice clean promoter region, or it looks differently. So the RNA or DNA polymerase doesn't know where to bind to that gene to get started. So what they'll do is they will take a strip. So here's our plasmid. The scientists know that the promoter in our plasmid is like right here, and that is a normal bacterial promoter. They will cut the plasmid such that the new DNA that they are sticking in goes right after that promoter. So when the machinery from the prokaryote latches on to the normal promoter that it is used to, it'll just continue down the rest of this line and copy the rest of the gene that's along with it, so they don't have to really worry about getting things going. That's just one strategy. There are many. Keep that one in your head for use later on. Next thing to talk about, like I said, we got a bunch of stuff to cover today, is the polymerase chain reaction. This is one you will hear about a lot, and this is a very commonly used lab technique. If a scientist has got, let's say, a small fragment of DNA, but they need a ton of copies of it for research, because it's hard to research on just one tiny little fragment. You need a ton of fragments. What they will do is they will take their DNA that they've got, and they go through this process of heating and cooling with enzymes present. So the first thing they do is they take their DNA strand and they heat it up briefly, and that separates our double helix into two ladders. They then dump in necessary RNA primers so that the replication process can happen. Those RNA primers and the proper ligase binds things together, puts our primers down. They then put in some DNA polymerase on the proper bases. DNA polymerase goes to work and it hooks everything together. They give it some time to cool and re-solidify, and there you go. You now have got new copies of DNA, and then you just continue this process over and over again. You heat it up, throw the bases and the enzymes in, let them do their work, let it cool down, and kind of come back together, heat it up, and you can do this continuously until you've got enough DNA to do whatever research you need to do. And then as far as analyzing DNA goes, there's a process called gel electrophoresis, and it sorts DNA fragments according to their size. Now, we've talked about enzymes quite a lot. Depending on the type of DNA, the same enzyme will make different fragments. So let's say this is DNA from person A, this is DNA from person B. You dump in the same restriction enzyme, A might be cut into 3 pieces, B might be cut into 2 pieces, or you could say B is cut into 3 pieces, but 2 are big and 1 is really tiny. What scientists will do is they'll take their different samples, they will cut it up using a restriction enzyme, and then there is a gel that has got little wells in the end of it. They'll load each of the samples into a well and pass an electric current through it. Now, here's the cool thing. DNA is negatively charged, so if you put the positive into the current down here, DNA will automatically migrate towards that positive end. But the thing is, this gel acts like a sieve. It like sorts things out. It's got little tiny holes in it that the DNA can travel through. So the further down the DNA fragment makes it, the smaller the DNA fragment because it's able to fit through further. The higher up it stays, the bigger the fragment. So you can see over here on our picture, here's the wells. This band right here represents a really big DNA fragment because it couldn't travel that far. Smaller fragments would be all the way down here because they were able to travel further down the gel. Now, because 2 samples of DNA are different, they will cut differently, which means you will see, like we see over here, different patterns. Like these 2 right here, they are very similar or identical DNA. You can see their banding patterns are pretty much the same. But if you look at these 2 right here, their banding pattern is completely different. So this can be used to compare a lot of different DNA sequences together. Last thing, hooray, we are there, southern blotting. For southern blotting, this is another technique that scientists use to identify fragments of interest or DNA of interest. It does use the idea of nucleic acid hybridization. So what a scientist will do is, using electrophoresis, they will cut up their DNA, they will separate it out into fragments. The nice thing is, once you run it through electrophoresis, each one of those bands is a chunk of DNA fragments that is the same. So you've isolated your DNA and put it in little chunks and packets. But you still might not know which one of those packets has got the genetic material you need in it. So what they're going to do is they've got their gel that they just ran electrophoresis on. They put it into a salt solution with some filter paper and a sponge on top of it. It's a high-tech setup, let me tell you. So the salt solution passes up through the gel, it passes through the filter paper and into the sponge. As it passes through the gel, it takes the DNA along with it. And all that DNA is transferred here to the filter paper. So all your DNA that was in the gel is now in the filter paper in the same pattern it was in. Next thing you do, you put together a nucleic acid sequence that matches the gene that you are looking for, and you label it with a radioactive phosphorus. Stick all that in a bag, and those radioactive phosphorus that you built are going to stick to the gene that you're interested in. Pull it out, get rid of the probe, and last thing you do is you will throw some X-ray film over the top of your filter paper that now has got the radioactive probe on it, and it will leave marks on the film because that radioactivity exposes the X-ray film. So we know that we started out with this, you can see all the bands, and at the end we know the genes that we wanted are right there, and right there. So scientists can then go back to their filter paper, recover that DNA, run it through PCR, and make enough copies of it for whatever they're trying to do. I know that was a lot of junk. Forgive me for the bad edit in the middle of it. Thank you for hanging out. This has been the Lab 207 webcast. We'll see you again.